Good morning, everyone. So, in the past class, we already discussed about the biochemistry of iron, magnesium, etc. And now, in this last session, this is the fourth part of bioinorganic chemistry. And in this session, we would like to discuss about biochemistry of certain metals such as zinc, cobalt, etc. So, first, let's discuss about biochemistry of zinc. So zinc has important biological role and it is mainly present in some enzymes. So in a human body about 2 gram of zinc can be found, and it is the second abundant transition metal in human after iron. So there are more than 8 enzymes identified which contain zinc, and among them some are discussed. The first one, carbonic anhydrase. It is present in RBC and involved in respiration. So it speeds up absorption of CO2 by RBC in muscles and other tissues, and also reverse reaction involving release of CO2 in the lungs. So in the past class we discussed about oxygen transportation cycle, oxygen carbon dioxide transportation cycle. So in that particular cycle there is a enzyme involved called carbonic anhydrase. Now second one is carboxypeptidase. It is present in pancreatic juice. Its role is digestion of protein. Then alcohol dehydrogenase, the third enzyme that contains zinc is alcohol dehydrogenase. It is involved in alcohol metabolism. So it oxidizes alcohol into aldehyde and reduction of aldehyde to alcohol. So it is actually involved in the metabolism of alcohol. Then dehydrogenase and aldolases. So these two enzymes involved in sugar metabolism. So there are several sugars, disaccharide, trisaccharide, something like that, and these enzymes reduce them, hydrolyze them into simpler sugar units. Then alkaline phosphatase. They are involved in certain energy releasing reactions. So they related to ATP. Okay, when they interacts with ATP, energy may be released. Then biochemistry of cobalt. So cobalt is one of the essential trace metal. Number of enzymes contain cobalt which are referred to as cobalamin complexes. So the cobalt containing enzymes are generally called cobalamin complexes. And we have 3 different types of cobalamin complexes, or we can say that precisely there are 3 different types of enzymes that contain cobalt as central metal. And first one, cyanocobalamin. Second one, adenosylcobalamin. Third one, methylcobalamin. All together we call them as cobalamin complexes. The cobalamin complexes contain hexacoordinate cobalt 3 ion at the center of a coring ring. See, similar to porphyrin ring, there is another one called coring ring. So it is a hexacoordinated cobalt 3 complex, and it is placed at the center of a coring ring. The cobalt is bonded to 4 nitrogen atoms. See, in hemoglobin, we could find a porphyric ring. Similarly, in cobalamin complexes, you can find a coring ring. There will be a coring ring. So here the cobalt is the central metal. Its oxidation state is 3 and 4 nitrogen atoms are surrounded, and the 5th position is occupied by another hydrogen from a side chain. Okay, as in the hemoglobin. Then the 6th position is occupied by a cyano group in cyanocobalamin. So that's the difference. Only the 6th valency is different in the case of cyanocobalamin, methylcobalamin, adenosylcobalamin. So in cyanocobalamin, 6th position is occupied by a cyano group, and in adenosylcobalamin, an adenosyl group occupies the 6th position. Similarly, in methylcobalamin, a methyl group occupies the 6th position. So the human body contains 5 mg of cobalamins, and lack of cobalamin causes pernicious anemia. Vitamin B12 is essential for making red blood cells. It also maintains the nervous system and required for many enzymatic transformation. Glutamic mutase enzyme, which is involved in the metabolism of amino acids and ribonucleotide reductase, are adenosyl cobalamin dependent enzyme. Then methylcobalamin is related to the methyl transfer reactions in biological system. See, while we are concluding the actions of these cobalamin complexes, they act as some sources. They are some sources in the body for the for the supplying of certain groups. So in the case of methylcobalamin, they are involved in methyl transfer reactions, and in the case of adenosyl cobalamin, they involved in glutamic mutase enzyme, which is involved in the metabolism of amino acid. So they have their own particular duties, and their deficiency may cause pernicious anemia. So keep in mind, their deficiency cause pernicious anemia. Then we are moving to the last of the chapter called the anti-cancer drug. So we have two drugs mainly. First one is platin and second one oxaliplatin. So in cisplatin, chemically it is cis-diamine dichloroplatinum 2. Its mode of action is, in the case of every anti-cancer drug, its mode of action is simple. It binds with the DNA. And it interrupts the transcription and the translation, so that the replication information to a cancerous cell may be blocked. So the cancerous cell won't replicate further. So for replication also it requires a message sender, a messaging from the DNA. So if it is blocked, if the message for replication is blocked, then it stops the unwanted multiplication of the cancerous cells. And this happens when anti-cancer drugs acts on our body. Then there are several compounds based on platinum that play a central role in many cancer treatments. Two of these are cisplatin and oxaliplatin. General mode of action. In general, platinum containing anti-cancer drugs bind strongly to DNA in region containing several nitrogenous base units, preferably guanine. They bind in such a way as to form links within each strand and bind between the strands sometimes. Transcription and replication are inhibited as a result. This eventually triggers cell death, and this is how it acts. First one, cisplatin mode of action. Cisplatin binds strongly to DNA in regions containing nitrogenous base units, binding in such a way as to form cross links within the strands. That's the main thing. So cisplatin binds within the DNA strands, not between. Okay, not between. Thus, cisplatin interferes with DNA transcription and replication, thus kills the cells. Cisplatin is useful in treating various types of cancers, including testicular, bladder, lung, esophagus, stomach, prostate, etc. Limitations: eventual development of resistance to the drug, as cancer cells uses all the mechanisms at their disposal to fight disruption by cisplatin. It causes side effects including renal toxicity, means kidney failure, neurotoxicity, and induces severe nausea and vomiting. So this is the structure of cisplatin. Then oxaliplatin. When we compare cisplatin and oxaliplatin, the oxaliplatin is found to be much better when compared to cisplatin. So its mode of action, platinum in oxaliplatin gets attached to nitrogen bases of DNA, the resulting intrastrand and interstrand. Keep in mind, the oxaliplatin creates both intrastrand and interstrand cross links. Intrastrand and interstrand. There is a correction. Please note it. It creates both intrastrand and interstrand cross links, interfere with the cell division and prevents DNA replication as well as transcription. This in turn causes cell death. Oxaliplatin in use is used to treat bowel cancer, means abdominal cancers, esophageal cancer and stomach cancer. Then advantages of oxaliplatin over cisplatin. See, as we said earlier, there are several advantages for oxaliplatin over the cisplatin. The oxaliplatin is associated with the comparatively reduced toxicity. Then its neurotoxicity is reversible and is thus it limiting in contrast to that seen with cisplatin, which is irreversible. So its toxicity is reversible. That's a simple thing. Then renal toxicity occurs only rarely in contrast to cisplatin. So basically, the toxicity of oxaliplatin is very low when compared to cisplatin. So nausea and vomiting after its administration are also minor. And this is the structure of oxaliplatin. Keep in mind. So these things you can expect question from anti-cancer drugs. So these things are very important. So that's all about the chapter. Thank you. Thanks a lot.